Hello, molecular biology. I hope you are all safe and doing fine and haven't gotten sick. Today we start our adventure of the flipped classroom. So this is going to be a more asynchronous model where you're going to be expected to read and review the material on your own, but you can do this at any time during the week that we're supposed to be reviewing it. So we are going to follow the syllabus. And then I'll be posting these weekly videos to highlight areas where you need to focus, things that are going to be due, and otherwise kind of help direct your study as we go through the material. So, before spring break, we finished the what was it? Transcription pre-initiation steps. We looked at a lot of different things that could affect transcription initiation and therefore the start of gene expression. And there were lots. We focused a lot on transcription factors and proteins and how the activity of all those things are regulated. For this next week, we're going to continue looking at post-transcriptional events. We got through most of the RNA processing slide. That's slide number 15 in the files on Canvas. We looked at a few of the modifications that occur only in eukaryotes, remember. And then the general splicing mechanism. You don't need to know specifics. You just need to know the power of splicing, that you can get multiple different types of RNAs from one gene, and that's going to increase the diversity of the proteins the cell can make. So that really is the power of what's called alternative splicing. Okay, for the week of March 23rd, we're going to start looking at other roles that RNAs have in regulation. So slides 16 and 17, they talk about microRNAs, RNA interference, long non-coding RNAs. There's lots of different RNAs that can have an effect in the regulation of gene expression. You want to focus particularly on microRNAs and the fact that we can use microRNAs as well to generate what's called a knockdown of gene expression for a particular gene. So you want to take a look at how RNAs are used. There's things like riboswitches, and then pay particular attention to microRNAs, the mechanism of microRNAs. The paper has to do with what's called suppressor genetics, and they're going to use a microRNA screen to get information on different genes within the cell. Back in the old times, people would do what's called forward genetics, which is where you would use a mutagen or something like that to make random mutations throughout the genome. And then you would screen the mutants and look for cells or organisms that gave you phenotypes of interest. And then you would have to go back and find where you made that mutation and then identify the gene involved. MicroRNAs and RNA interference can be used in what's called a reverse genetics manner. So you can actually target particular genes, or one gene, or groups of genes, or the entire genome, and then you look for phenotypes of interest, but then you can quickly identify whatever genes you've targeted. So it's a more, you know the targets that you're already targeting rather than starting from the forward genetics. So that's reverse genetics, is using microRNAs, for example, to target specific genes and then look for phenotypes of interest. But then you kind of have an easier job at figuring out what genes that you were targeting to begin with. So focus in on that, slides 16 and 17. 17 talks about suppressor screens. This is another way that you can find proteins of interest within a pathway. And that's pretty much the major things from those. That's what we're going to focus on this week. Things that are due, looking at due dates, I did extend the transcription paper due date, so you want to get that churned in. There's another, we're going to be doing the suppressor paper, so that's going to be another paper write-up that'll be due later in the week. And you have a Smart Work Chapter 19 that's due on the 28th. And then I've also been working on the background sections for our grants. So you can start working on the methods section for your grants. So this is where you kind of spell out the methods that you're going to use under each of your specific aims. Again, you don't have to go to the level of listing PCR ingredients, but you do want to say, describe the kind of PCRs you're going to use, for example, or if you're using novel techniques, you definitely want to discuss those. So that experimental methods will be due later in April, but you want to get started on it. And then I'll be sending feedback on the preliminary background sections for you to work on as well. So I think that's it. Please leave comments in the comments section for this video or email me with questions as you go through and review the material. Oh, by the way, I forgot to mention, there are audio files available on Canvas. So for this flipped classroom, you can listen to those audio files. You can download them to your own Dropbox or just listen to them within the browser. Follow along in the notes, in the PowerPoint slides as you go through those audio files, and then generate your questions from that and send them to me so I can clear them up for you. Okay, good luck and take care. See you later.